introduce the very first speaker, Jan Johansson. Dr. Jan Johansson is president and CEO of ARTERY Therapeutics. He's a serial biotech entrepreneur and has, as founder and or corporate officer, helped to take 3 companies public and raised more than $400 million in private and public markets. Jan is trained as a basic scientist, practiced cardiovascular medicine for 18 years and has translational and development background with particular reference to cardiovascular technologies. In the CVD space, he led basic research groups and clinical phase 1 to 3 programs. He was also responsible for an NDA product registration, Amacor or Lovaza, and he's published more than 60 peer-reviewed articles and more than 100 abstracts in the CVD area. And he's the inventor of more than 30 patents. And today he's going to tell us about Cogpep for the treatment of ApoE4 associated Alzheimer's disease. Jan.
Thank you. Well, thank you, and excuse me for making that so lengthy. It's wonderful that you can keep this meeting up and that so many people are coming and attending. So thanks to Syed, Chris, and everyone and the scientific crew. So the name of the company is ARTERY Therapeutics, and it started out that way because we focused on the ABCA1, that's a transporter of phospholipids and free cholesterol across cell membranes of monocyte macrophages. But it turns out that this technology has become of importance for Alzheimer, and particularly the ApoE4 Alzheimer's. ApoE4 is a lipoprotein, and the brain is full of lipoproteins, and lipoprotein transport in the brain is important. So we have, to a large extent, shifted our focus to ApoE4-driven Alzheimer. So I will tell you about our first couple of proof of concept trials and how we project that this is going to work going forward. So I'll read the title because it contains a lot. The novel ApoE4 Therapeutics, CS6253, penetrates the blood-brain barrier, which is unusual for a peptide, upregulates the ABCA1 transporter, and prevents Alzheimer's disease pathogenesis. Focus on treatment of homozygous E4. So if you look at the shift in death prevalence from 2000 to 2010, you can see the enormous increase in Alzheimer's disease, diabetes pretty much unchanged, decrease in cancer and heart disease and stroke. And with the higher prevalence of Alzheimer, you also have the higher cost. You can see here that the cost here now is about 200 billion per year for the US as a society, and to 2050 this is going to increase to about $1 trillion. So this is going to be the biggest burden on the society. When you look at Alzheimer's disease genome-wide association studies, you find that ApoE is the strongest risk factor described by Alan Roses about 15 years ago, but also other lipoprotein transport potential targets are of importance. ABCA7, clusterin or apolipoprotein J, and PCALM, phosphatidylinositol clathrin associated. So lipoprotein is important. If you have ApoE4, which you find in the if you're homozygous for E4, which you find in the population in 2%, then you run about a 10% higher risk of getting Alzheimer. If you are homozygous for ApoE4 and you do not have cognitive decline at age 60, you will get cognitive decline. So E4 is a very strong risk factor. So E4 is approximately a 300 amino acid peptide. It has a C-terminal that binds to lipids that has a lipid affinity alpha helix. This part is also responsible for the take-up of cholesterol from ABCA1. So in the case of E4, you have an arginine in 112 and the 158 position, and that's putting constrictions on the C-terminal flexibility and its ability to receive lipid from the ABCA1 transporter. And this is basically how it shifts a lipid-free ApoE interacts with the ABCA1 transporter to take up phospholipids and free cholesterol. So we have these licensed technologies from Berkeley, and we have designed peptides with the help of John Bailicki, but also we got some nice help from Valid. Thank you, Valid. And they are mostly class alpha helical amphipatic peptides. They are ABCA1 agonists. They are potent and selective, druggable and favorable safety profiles, and they cross the blood-brain barrier and prevent ApoE4-driven Alzheimer. And that's what I'm going to tell you about. First, if we look at cells, so in the brain, the macrophage cells are astrocytes and microglia. If you look at ApoE4 astrocytes and ApoE3 astrocytes before and after peptide co-incubation, you can see how the ABCA1 agonist CS6253 really increases the cholesterol flux across the cell membrane, about 8-fold. So what we did in a set of studies was that we established a model to test the in vivo efficacy. So ApoE4-driven Alzheimer is characterized by increase in amyloid beta, phosphorylated tau, synapse and nerve cell deterioration, followed by cognitive decline. And all these human Alzheimer's disease traits were reproduced in ApoE4 target replacement mice, where we have removed the murine, the mice ApoE, and inserted the human ApoE4 and E3. And then we have contrasted the E4 and E3. And we see that Cogpep, which is the name we use for CS6253, 100% counteracts the ApoE4-driven Alzheimer, both the phenotype in the brain and also the cognitive decline. And we have also, the background in these ApoE4 target replacement mice is a wild-type background, so we have only changed the ApoE, but we're also working with a second model in the second lab to feel comfortable about the results. And there the background is 5 knock-ins of familial Alzheimer's disease. So it's pre-senilin-1, it's amyloid precursor protein. So that corresponds to getting Alzheimer at age 5 or something. A terrible type of disease. So it's important to understand the pharmacokinetics. We did that by testing 20 milligrams per kilo sub-Q and IP, and then we gave it IV. So we see that we have a 70% bioavailability. The half-life in serum and brain is about 7 hours. We see that we reach the concentrations in the brain sufficient to match these 50 values. And very interestingly, in the hippocampus, we see a co-localization of the peptide with the astrocytes, which we would expect because the astrocytes have the ABCA1 transporter. So the proof of concept studies were done where we treated with 20 milligrams per kilo every other day. We treated every other day because we had done functional tests and saw that the peptide had effects longer than 24 hours. And then we had 4 groups, namely ApoE3 mice that received control, ApoE3 that received CS6253, E4 control, and E4 received active drug. This is the co-localization, simply showing here that where you see astrocytes, you see peptide, and where you see peptide, you see astrocytes. And this is 24 hours after administration, the final administration, and in hippocampus, the most important part. This is the pharmacodynamic effect, where you see that the E4 non-treated, they have a high concentration of lipid-free peptide, while after treatment, that lipid-free peptide is taking up lipid and is now found here with the larger species. So it's a particle gradient where the treatment effect is to have these guys here take up lipid and then migrate naturally become larger particles, and that's illustrated here. So when we look at the ABCA1 transporter, the protein concentration is increasing a fair amount. Excuse me. It's going up here. You see the E4 control treated mice with treatment. They increase the ABCA1 protein concentration increases about 50%. The peptide binds to ABCA1, prevents its internalization and degradation. When we look at cognition test, you can see that the real only outlier is the ApoE4 control. When they get treated, they assume the normal learning curve on a Morris-Water maze test. That's the most standard test. One part of that is the probe test, where you can see that the E4 mice normalize and behave just like the E3s. Also did a second type of novel cognition test called novel object recognition, and also there you see a total normalization of the cognition in the E4 mice that received treatment. And what explains this prevention of ApoE4-driven Alzheimer? Well, we see that amyloid beta is normalized, is becoming normalized. It's decreased with about 60%, similar numbers almost for phosphorylated tau using the standard antibodies. When we look at more specific synapse, and this VGLUT1 is a glutaminergic epitope, you see that it increases more than 100%. ApoE4 is short for ApoE receptor 2. That one increases, and it actually also increases in ApoE3. And DCX, double cortin, is a growth factor, which is increased as a compensation in the control animals, and it's now becoming normalized because it does not need to compensate any longer. So in short, when we look at the treatment effect, it's doing everything it should. It's, we see target engagement, we see drug at the right place, it has the neuronal effects and the effects on synaptic viability. I won't talk about it, but we've also figured out a good biomarker in plasma that we can use for following PK/PD. So this is in short how we believe it works. When it comes to the amyloid side, you know, in the Alzheimer field, some people are amyloid biased. Others feel that maybe we should look at some other things too. But the big companies so far are pushing antibodies for amyloids and BACE inhibitors. The current idea is that the soluble oligomeric amyloid beta is the toxic portion. And when you're able to lipidate ApoE with the help of the ABCA1 agonist, then you solubilize the amyloid beta and you increase its degradation. So we see in this familial Alzheimer's disease background mice in the other lab that we decrease soluble amyloid beta with about 60%, and we're very happy for that. But we believe that there are other effects as well. So in summary, ApoE4 Alzheimer's disease is a lipidation deficiency disease. And I didn't go into the details, but there is strong epidemiology the last 3 or 4 years for that statement. There is no treatment for ApoE4. As a matter of fact, the amyloid antibodies that Biogen, Roche, and Lilly are pushing have a high prevalence of side effects in the ApoE4 population, including encephalitis. So we seem to be totally alone in this very important group. An ABCA1 agonist, CS6253 peptide, can counteract ApoE4-driven Alzheimer. And from about 2024, there may be a need of 5 tons peptide per year. And these are the hardworking people, and she is by far the hardest working people. The PhD student who soon will defend her PhD in Tel Aviv, the inventor of the peptide, the PhD mentor, and Alois Alzheimer, the German guy who observed the problem. Thank you so much.